Sorry.
This is lecture 24. We're going to skip from the endocrines and kind of pick up the endocrinology of the reproductive tract. So I'm going to skip to plate 149. I get students in advising, should I be a biologist? Should I go into animal science? What's the best? And I think what's the best is really kind of a function of what you want to do and what you want to emphasize. And I've my undergraduate degree is in biology, my training in zoology, and it was then when I moved into animal science. And I think that both of the disciplines offer certain strengths. And I would have to say that one of the big strengths in animal science is the attention and detail and the ability to manage reproductive cycles. And a lot of what we know about reproduction came from our work in domestic animals. The sheep still serves as a primary model for human female reproduction. So this class is meant as an overview. We have an excellent reproductive physiology class. That's certainly one of the advantages, and you should avail yourself of that. I will simplify some things. I get into trouble on times because the theriogenologists teach in great detail, and I kind of smooth things over. I also skip a lot of the detail. You're going to spend a lot of time in your different classes talking about different cycle periods, reproduction. Frankly, I forget a lot of them. So I can't remember what's the gestation period for a cow. So some of these things I don't emphasize in this class. I want kind of the general overview. And we're going to start on plate 149 to look over an overview of the male reproductive system. Ultimately, what we want to produce is fertile sperm, because it is the sperm and the egg that are going to combine to make the outcome, whether it be a calf or a foal or a human fetus. But involved with that are a lot of other secondary sex organs. So the primary sex organ is the testes. But in addition to the testes, there's kind of an elongation or an enlargement of the testicular mass called the epididymis. So think of a tubular network coming from the testes in through the epididymis, and at the bottom here are the testes, and then there's the enlargement of the ductal tubes from the testes into this epididymis. Now here's a species difference. In our domestic species, in the ruminants, the epididymis is at the dependent portion, but it's kind of an enlargement of the testicular mass. The tubules from the epididymis feed into the vas deferens. The vas deferens then feeds into the urethra. So there's a common duct, a urogenital duct, that delivers not only urine but also material fluid, which is produced in the testes and modified by these secondary sex glands. So in addition to the testes, we have glands located along this, including the seminal vesicle, the bulbourethral gland, sometimes called Cowper's gland, and the prostate. And we mentioned the prostate as being one of the sources for prostaglandin, so we're going to come back to that. Female. The testicular equivalent, that is the one that produces the sex steroids, is the ovary in the female. The ovary is connected to a smooth muscular uterus by a uterine tube, sometimes referred to as a fallopian tube, and then that exits into the vagina, so that the vagina then is the commonality for the urogenital system of the female. So here is If you can take me out of the picture for just a second. Here's the uterus. Here's the bladder, and you can see that the entrance into the uterus is the cervix, and then you have the common vagina. Secondary sex characteristics in human males, dogs, cats. In males, in humans, we recognize facial hair as being, or hirsute, body hair, as being a component. But in addition, muscling, thicker muscling under the influence of testosterone. Vocal cord changes, particularly in humans, we recognize voice pitch. Aggression. There's no question. A lot of testosterone, a lot of aggression. Female, estrogen-like secondary sex characteristics. Estrogen is driving development of the reproductive organs, development of the mammary glands. Males have mammary tissue. Males can get mastitis. They can get inflammation. Not as common as females because the tissue isn't as large. Lack of facial hair, lack of body hair, more fat deposition. So more subcutaneous fat in females, important in energy storage, particularly when you're thinking about carrying a fetus. So these are commonalities shared by all of our domestic animals. So let's look at let's kind of look at each system independently. We're going to start with the males because it's the most boring. Sorry, gentlemen. It's much simpler. And let's look at it histologically. So here's a nice diagram that's kind of a cartoon that shows you that the testes is actually composed of lots of these wiggly little tubes called seminiferous tubules. The seminiferous tubules, then, if you take out and look at this tubule, you'll see that it has a central lumen, a duct, if you will, that transports material, primarily semen, down this duct that then feeds into the epididymis. So this is the epididymis. This is a convoluted tubules, which are extension of all of these seminiferous tubules. They dump then into the vas deferens. And so there's kind of the enlargement right there. So let's look then at the detail of these seminiferous tubules. And what they've done is they've taken a nice enlargement right here so that this space represents the lumen. And these cells right here sit on the opposite side of this basement membrane. So you can think of these kind of as like garden hoses. This is the inside of the garden hose. This is the wall of the garden hose. And you've got a lot of these garden hoses or seminiferous tubules that are stuck together. And in the spaces are cellular material. Two major types of cells. The ones that are noted here in kind of yellowish orange are called Leydig cells. It's these cells that are responsible for the production of the male hormone testosterone. The testosterone is produced by these cells. It goes into the general circulation for distribution to all of the tissues. It's also exported locally for its influence on the cells involved in sperm formation. There is a basement membrane on which sit the spermatogonium. So now you know a spermatogonium, that's like a precursor for sperm production. Under the influence of testosterone, these cells undergo division, mitosis and meiosis, right? Mitosis, cell division, meiosis, going from a haploid or from a diploid to a haploid cell. So what you need to do is you need to generate sex cells that only have one half of the chromosome complement. This process occurs from the basal membrane outward towards the lumen of the duct. So cells up here, spermatids are young sperm. So they've got tail development, but they're not fully mature. So we go from a spermatogonium to a spermatid. Cells that are up here are older than cells down here. Cells up here are haploid. Cells down here are diploid. They need testosterone in order to undergo spermatogenesis, or sperm formation. So no testosterone, no sperm formation. But they also require the involvement of a supportive cell found within this progression of maturing sperm cells, and that is the Sertoli cell population. The Sertoli cell population nurse or nutrient cells. They help support this process and maintain the viability of the sperm from its spermatogonium to the spermatid form. It does this in part by elaborating a protein called androgen-binding protein. URL not there, so work. Nope. Well, I'll do it up here. Androgen binding protein. It's a protein which does what? It binds androgen. The primary androgen is testosterone. That means that this cell is producing androgen-binding protein that picks up this testosterone. It binds this androgen and keeps it in close proximity. So local concentrations of testosterone are very, very high within this region. A concentration that is necessarily high in order to have spermatogenesis. So if you are not producing adequate numbers of sperm, you can look to a testosterone deficiency or maybe a failure in the Sertoli cell population. Factors that influence sperm formation. One of the biggest is temperature. In order for this process to occur, it tends to be at temperatures that are below core temperature. As a consequence, we find that in most of our domestic animals, the testes sit outside the abdominal cavity. They hang in a very vascular, cooled sac. So there's a scrotum outside of the abdominal cavity. Animals that have retained testes are oftentimes infertile because body temperature is too high to support normal sperm development. They have abnormal, non-functional sperm. In humans, right, problems identified with cyclists, spending too much time in the hot tub. What do you think the number one reason for infertility might be in our domestic animals? What do you think the number one cause of elevated temperatures might be? Certainly, it's hard to picture the dog in the hot tub or getting to ride a bicycle, but what about fever? So they get an infection, generalized elevated body temperature. That gets translated down even into the scrotum in some cases, where it can inhibit and kill off maturing sperm. Now, short of radiation, you seldom kill off the spermatogonium. So these cells, they won't undergo appropriate division if the temperature is too high, but they don't get killed off. So these cells, they die when the temperature is too high. That means that after a fever has broken, or after you removed a thermal insult, it takes time for it to go from spermatogonium to spermatid. This process is the time of this process can be quite protracted. It varies from species to species, but may be on the order of 50 days. Meaning that if you had a fever and you killed off all your sperm, it may be another 50 days before you're fertile again. Other causes, toxins, and the important one pointed out in your notes in the plate is gossypol, cottonseed, common feed supplement to cattle. Give them too much gossypol, become infertile. Bad news if you're raising cattle for money, right? And then we made a comment about radiation. One of the side effects of chemotherapy and radiation therapy is destruction of the sperm cells, and that includes the spermatogonium. And once those have been eliminated, you no longer have a regeneration. So if you kill all of those off, you're no longer going to be fertile. One of the reasons that they tend to do testicular harvests before chemotherapy. Last one mentioned, vitamin E. Vitamin E important in a number of disease processes, important in skin function, important in immunology, also critically important in reproduction. Inadequate levels of vitamin E lead to infertility. If you look in domestic animals, the NRC feeding requirements for our domestic animals, a huge focus on vitamin E requirements. Sperm maturation takes place in the epididymis. We have spermatids that are located right at the ductal face. These spermatids then swim up, and maturation occurs in the epididymis. Number and characteristic of sperm cells, I consider that important in a repro class. Numbers do change as a function of species. That number is reflected not only in concentrations, the number of sperm per mil of ejaculate, but also in the volumes. And a lot of managed breeding has to do how much semen can I get, how much can I thin it out, how many aliquots can I produce, how many inseminations can I get off a single ejaculate. So the numbers here are all specific to humans. Semen function. Plate 151. We need to have mature sperm, and we need to find plate 151. So if you have to give me a second here. Oh, this is my CAT scan. You like this? A CAT scan of a hip on a horse. Plate 151. Well, it seems that I didn't scan it. So can we go to the overhead on this one? Always adapting, always making the sense. So we need to not only have functional sperm, but we need to have a fluid matrix that supports that. And a lot of that fluid comes from accessory sex glands. So we have semen, which is in a fluid which is referred to as seminal fluid and seminal plasma. This fluid comes from the seminal vesicles, from the prostate, and from the bulbourethral gland. One of the primary elements, so here we've got vas deferens, seminal vesicle, prostate gland, and the bulbourethral gland. These are all adding components into the semen to produce this seminal plasma. This includes alkaline secretions, so it tends to be a basic solution. It has, in part, to neutralize the acid in the vagina of the female. So you're trying to make an environment that is supportive for the sperm. Prostaglandins. Prostate dumps in prostaglandins. The prostaglandins are important in uterine contractions to help move the sperm up the uterus, up the uterine tubes, up the fallopian tubes towards the released oocyte. Some vitamins, nutrients, amino acids, energy sources, glucose, fructose. So a supporting matrix, as we'll look in more detail, the sperm consists mostly of mitochondria and not a lot of cytoplasmic tissue. That means it does not have a lot of nutrient storage. It's only available and able to be functional for a relatively short period of time. So you need to get the seminal fluid in there, and you need to get the sperm. So you need to have erection. Parasympathetic nervous system is involved in modulating vasoconstriction of the vasculature in the penis, so that erection occurs under parasympathetic activity in which blood flow is increased to the tissue, to the interstitial tissue, and the penis is characterized by having spongiform tissue, a lot of sinuses. So the erection occurs through the swelling, the movement of blood into the penis. So you now have an erection, so you now have a device that can insert into the female. In our domestic animals, damage to the penis, breaking, hematomas that causes deviations and changes in its shape, makes the animal non-functional. So when I go out and do a breeding soundness exam on the bull, I need to ask the question, does this animal have a normal erection? And are there any adhesions? Is there any problem with the animal, with the penis coming out of the sheath? Is it normally shaped? A characteristic surgical treatment, veterinary surgical treatment, is penile deviation. If you want a bull used to identify cows in heat, great to have a bull. Bad, the bull needs to have the testosterone to be interested. It's got to have the libido. But you don't want him inseminating each of the animals. You merely want him to identify by jumping up on the back of that female. So how do you do that without getting the animal pregnant? You alter the directional flow of the semen. And so there is a surgical procedure in which you angulate or bend the penis at a 90-degree angle at the end so that these animals aren't able to insert or intromit the penis into the vagina. So you've got to have the erection. If you know that this is modulated by nitric oxide, do you remember us talking about it as a vasodilator, a compound that was produced in metabolizing skeletal muscle? Well, it also plays a critical role in vasodilation of the penis. And if you're a pharmaceutical firm and you come up with a compound that supports nitric oxide production, so it inhibits the enzyme involved in the breakdown of nitric oxide, you can have penile erection. And we now see a multi-billion-dollar market for Viagra and its role in erectile dysfunction. We recognize it in humans. We recognize it in our domestic animals. So we have the parasympathetic reflex response. Ejaculation. So we've got the penis erect. Now we want to deliver the seminal plasma and the semen. We need to contract the smooth muscles associated with the male reproductive tract. That is under sympathetic control. So the release of norepinephrine then manifests itself at the second phase in producing contraction and emission. Human sexual response has four phases, and it's not important that you recognize that. What you need to recognize is that you have important inputs from higher centers: olfactory, auditory, and optical input that feeds down and puts into this arousal process. So an important component of the arousal process is having those inputs. So there I am. I normally collect bulls by electroejaculation. That's because they are beef bulls, so they're not. They are seldom collected naturally. They are. There's much more AI in the dairy industry. So our bulls are collected primarily by electroejaculation. If that fails, I have in the past tried manual, which is collecting with an artificial vagina. Well, this is the classic example of having some optical input. So there was the bull. We had a cow that was in heat, so she was standing, she was receiving, she was sending off pheromones. The bull was getting an erection. He would start to mount her, and then I would move up to her and to interpose myself between her and his penis with an artificial vagina, and the visual input of having somebody in coveralls and big rubber boots coming at you was a distraction. So we're all aware of the importance of visual cues. Certainly. You see mares standing for estrus. They use a lot of visual cues. Same with our cows. And olfactory, pheromonal kind of transfer becomes very important. And so these can all modify the arousal response. So how does testosterone act? So if we turn to plate 152, which I hope I can find now. We need to have our testosterone involved. Hypothalamus. Hypothalamus produces gonadotropin-releasing hormone. Gonadotropin-releasing hormone has its impact on the anterior pituitary. The anterior pituitary releases. Do I have this? Oh, yeah. If we look at the diagram to the right, so there's emotions, visual input, auditory pheromones, releases GnRH from the hypothalamus. Anterior pituitary releases luteinizing hormone and follicle-stimulating hormone. Now, on the one hand, it's easier to start with males because it's a simpler system. However, both of these gonadotropic hormones were named from their function in the female. The stimulation of follicles in the female, and the production of a yellow body that we're going to talk about when we talk about female reproduction, which is the definition of luteinizing. So luteinizing means to make yellow. In the female, this hormone produces a yellow change in the ovaries. It's exactly the same hormone in the males, but instead of acting on the ovaries, it acts on the testes, and specifically, luteinizing hormone acts on the interstitial cells, which are the cells of Leydig, and you recall that these are the testosterone-producing cells. So there is a testosterone, a steroid hormone, that is under the control of luteinizing hormone, a protein hormone, and GnRH, which is also a protein hormone. So remember, hormones, releasing hormones and inhibitory hormones from the hypothalamus are protein or peptide derivatives. Same for the anterior pituitary, but now we see a steroid hormone, testosterone, being produced. FSH is also under the control of GnRH. FSH supports Sertoli cell function, androgen binding protein. Sertoli cells also produce a hormone called inhibin, and it is inhibin that has a, Nick, can you take me out of the picture just for a second? This little dash line, there you go. So in response, the Sertoli cells are producing androgen binding protein. It goes back and it inhibits. So this is negative feedback by a hormone, by another specific hormone, whereas the negative feedback from testosterone is more of the classic. That is, this tissue, the hypothalamus and the anterior pituitary, are responding to negative feedback by the hormone itself. In the case of inhibin, you've produced another hormone by the Sertoli cell that goes back to inhibit. You can bring me back. Now, the net effect is a pulsatile change. So you have pulses in GnRH, you have pulses in LH, FSH, and pulses in inhibin and in testosterone. And we're only beginning to look at the importance of the magnitude and frequency of these pulses in modulating the sexual response. It becomes even more important when we talk about the female in terms of synchronizing and making sure she's receptive and available. Okay, so the molecular base, I want you to know the molecular base for testosterone is cholesterol. So we know that we're dealing with a lipid moiety. The identification of target cells for testosterone are based on intracellular receptors. Where are they? Follicular cells, hair cells, so we start looking at the characteristics. Muscle growth. We talked about catabolic functions. Well, now we're talking about building or anabolic. So muscle growth. Bone growth. Bigger bones. Bigger muscles. You can use that strategy. Well, go out and look at a beef bull. Big. I mean, huge amount of muscling. Much larger. If you don't want that animal for breeding purposes, what do? Castrate it. Remove the testosterone. The animal's less muscled, less masculine. What if you do that to a human? If you want a male that's not particularly robust, and particularly one that doesn't have an interest in females, you castrate them. And if you castrate them early, you're not going to get these secondary sex growth. So the individuals will have smaller bones, smaller muscles, smaller vocal cords. The castrati, right? You wanted a male who had a high voice in his entire life. Well, take the testicles early. If you want somebody that's fairly bulked up but has no longer any interest in reproduction, castrate them later in life. Sometimes you can use that to your effect in domestic animals. You want a horse, you want it well muscled. You allow the testosterone to have some influence, and then you castrate them later. So again, from a production standpoint, you may not want any of that secondary sexual characteristics, and so you castrate them very early in life. Horses, you might want more muscling. You might want a bigger animal, so you allow the testicles to hang around for a longer period of time and exert their secondary effects. Okay. Closure of the epiphyseal plates. Bone growth becomes, turns out you need testosterone to initiate closure of the plates. So one of the consequences is you're getting larger bones, larger muscles, but also when testosterone tends to peak in puberty, bone growth stops. So you still need that pulse as well. All right, I think we're only at 35 minutes, but we've done male reproduction, and I'm going to save female, the more interesting aspects of female reproduction till the next set of lectures.